Hello and welcome back. So this is the second video related to microbial pigments. In this video we will be talking about prodigiosin pigment, isolation and the viva question related to this particular practical. Right, so in MSc you have this practical for pigment extraction, bacterial pigment extraction, and widely studied pigment that is prodigiosin is used in this practical. So Serratia marcescens is the isolate or it is the bacteria that is used for this particular practical. Right, so we will be studying about this example and the protocol. So let's start. So microbial pigments, these are the natural pigments which are extracted from microbes and hence it is termed as microbial pigments. Pigments are considered as secondary metabolites. They are not produced by each and every organism. Okay, so a number of different kinds of pigments are produced by microbes and are studied for their potentialities. Pigments are biodegradable, non-toxic and non-carcinogenic. Some textile, cosmetic and food industries are now producing and using microbial pigments on commercial scale. These natural colorants, they help increase the market value of that particular product. Like for example, um, nowadays people are demanding more for organic vegetables, right? So this is another example related to the pigments, right? So in case of synthetic dyes, the demand is increasing for natural pigments, and microbes are also a good source of natural pigments. So microorganisms, especially some bacteria, are nowadays used widely for commercial scale production of microbial pigments. Okay, so prodigiosin as a pigment. Okay, so the pigment we are going to study is prodigiosin, and in general the pigments, they are known to have properties like anti-cancer, anti-leishmania, then antimicrobial, antioxidant. These natural pigments, the lead or they are leading the market demand over the passage of time. And these natural colorants and pigments are derived from many different sources such as insect, microbes and plants. So among these sources, microbes and bacteria in particular, they have the capacity to synthesize a wide range of pigments. Why? Because of simple culturing techniques, pigment extraction techniques, then scaling up and being time economical. Microbes are used widely, and one of the example is Serratia marcescens, right? So this is the flow chart. I had mentioned this in the part 1 of pigment microbial pigments. You can refer there also. This is related to the importance or applications of bacterial pigments in taxonomic significance, functions and industrial applications. Okay, so moving further to our example. So Serratia marcescens is known for producing a red color pigment that is prodigiosin. Okay, so it is a motile gram negative bacteria and it is known for the production of prodigiosin. The other bacteria known for producing this particular pigment are Vibrio, Pseudomonas, Streptoverticillium and Alteromonas. The production of pigment prodigiosin has been observed to be affected by several environmental factors: inorganic phosphate availability, then media composition, temperature and pH. Addition of fatty acid components in media help increase the pigmentation of this particular bacteria. Okay, so prodigiosin is widely studied and it is widely used as it has this good properties like antibacterial, anti-cancer, anticocoidal, anti-malarial, antifungal, etc. Even prodigiosin is widely used in food industries, uh, pharmaceuticals and in textile industries also. Okay, so this is the protocol for pigment extraction. This is the in general structure of the protocol. So you grow Serratia marcescens on La, that is Luria bertini agar, and it can be added or it can be mixed with glycerol, or you can grow Serratia marcescens on milk agar as well. You should have a matte growth of the pure isolate. Then you add saline, sterile saline to the plate. You scrap the growth and you collect that in a centrifuge tube. You centrifuge the cell mass and you discard the supernatant if clear. Okay, otherwise you repeat centrifugation. This will give you cell mass. Now you need to digest this particular cell mass using one normal NaOH that should be twice the volume of your cell mass in boiling water bath for say 1 hour. Okay, then add equal volumes of ethanol. Okay, so now you had digested the cell mass, so the pigment that is produced by the cell is out of the cell. Okay, as cells are lysed, the pigment is out of these cells, so you add ethanol to solubilize the pigment. Now again you centrifuge and you get rid of the cell debris. Okay, so now to the ethanol you add equal volumes of petroleum ether. Okay, so now petroleum ether will help to precipitate the pigment. Okay, and now allow you mix the tube and you allow the tube to stand for 10 minutes and collect the upper layer. Okay, so now the upper layer will have the pigment. So if you want the pigment in liquid form with solvent, then you use it as it is, or you can further go to drying that particular pigment. Say for example here prodigiosin, you can go for evaporation of the solvent and you will get the dry pigment form. Okay, so this is the protocol for pigment extraction. So now let's answer some questions. Okay, so with respect to the practical and the viva exam, these are some questions that I think can be asked. Okay, so the first is, what is pigment? So the natural coloring matter obtained from microbial, animal or plant tissue is known as pigment. A pigment is a material that changes the color of reflected or transmitted light as the result of wavelength selective absorption. Okay, so this is the answer for what is pigment. Now, what are the applications of microbial pigment? So if you are asked this, then you need to give at least 4 applications, that is used in textiles for dyeing the fabrics, in food industries you can say for uh, food colorant, it is used as food colorant in ice creams, then baby food products, food supplements, okay, or confectionaries means some sweet dishes like chocolates. Then next is in cosmetics. Then another example, that is candle or wax industry, paint industry, in therapeutics, in biosensor or as feed supplement and diagnostic markers. Any 4 of these are sufficient for answering applications of microbial pigments. Okay, for Viva, I am talking here about Viva. Then can you name some pigments with their properties? Okay, so here you need to name the pigment and its properties. For example, pyocyanin blue has antimicrobial properties. Violacine has antiviral, anti-cancer, antiprotozoal property. Riboflavin is a good vitamin source. Then prodigiosin has antimicrobial and anti-cancer properties. Okay, so these are some examples I had listed. You can go for any other examples also, that is okay. There are many examples of pigments and their properties. Okay, so the one which is easy to remember for you, you can answer that. Then characteristics of microbial proteins. Okay, so microbial, sorry, not proteins, pigments. So microbial pigments are important source of natural colors. Okay, so this is very important that these pigments are not synthetic, they are natural source, so they don't have some negative uh, effects on environment or humans. Okay, so some are water soluble and some are not. Then molecular oxygen is essential for pigmentation, so only aerobic or facultatively aerobic microbes can produce pigments. Chemically, pigments are pyrrole, phenazine, carotenoid, xanthophyll, and quinone or quinine derivatives. Variety of pigments are produced by microbes which possess properties like antimicrobial, anti-cancer, anti-malarial, antioxidant, etc. So pigment synthesis is also dependent on pH, temperature, light, and media constituents. Okay, so these are some characteristics of microbial pigment. You can name at least 4 of these. Next, give some examples of other prodigiosin producing microbes. Okay, so at least two other than Serratia marcescens you can name, say Vibrio, Pseudomonas, Streptoverticillium, Alteromonas ruber, any of these. Okay. Next is give two examples of pigment producing bacteria, yeast, mold, or algae. Okay, so bacteria, there are many examples. I had listed the easy one here to remember. So the example is Pseudomonas, pigment is pyocyanin, then Staphylococcus aureus, zeaxanthin. Then in case of yeast, Rhodotorula, the pigment is Torula rhodine. Then Fafia rhodazim, sorry, Fafia rhodozyma, the pigment is astaxanthin. Then in case of fungi, you can name Monascus, for example, Monasco then Penicillium chrysogenum, pigment is chrysogenin. Then in case of algae, Dunaliella salina, the pigment is beta carotene, and in case of Haemococcus, again astaxanthin. Okay, then next question is what are the characteristics of prodigiosin? Okay, so the prodigiosin, it is a pigment, and we know that pigments are secondary metabolites. So you can say the prodigiosin is a secondary metabolite. It is red in color and lipid in nature. This is very important. Prodigiosin is lipid in nature. Okay, and that's why we use petroleum ether for the extraction. Okay, then it is produced intracellularly and it has antimicrobial, anti-cancer properties. Then next is why glycerol broth or milk agar is used. Okay, so answer to this is fatty acid substrates are known to stimulate pigmentation, and that is why we use glycerol broth or milk agar for pigment extraction. Then what is the role of ethanol in the protocol? So it solubilizes pigment, and petroleum ether, it precipitates the pigment. Okay, that's why we use ethanol and petroleum ether in the protocol. Then next is what is the mechanism of action of prodigiosin. So according to the research, there are four possible mechanisms which are suggested attributed to the prodigiosins are pH modulators, then cell cycle inhibitors, DNA cleavage agents, and mitogen um, activated protein kinase regulators. Okay, so these are some um, mechanism of actions of this pigment. Then what are characteristics of Serratia marcescens? So it is a gram negative, sorry for the mistake, G should be capital. It should be gram negative, non-motile, uh, motile and citrate positive bacterium. The colonies on nutrient agar are convex circular with entire margin. Okay, so you can say at least 3 to 4 characteristics of Serratia marcescens and it produces the red pigment. You can add that also. So why do bacteria produce pigment? This is also a very important question. So pigment formation is associated morphological characteristics, cellular activities, protection for pathogenesis and survival in stress conditions. For these particular reasons, bacteria, they are known to produce pigment. Okay, so what are the advantages of these microbial pigments? Why microbial pigments? Okay, so they have broad range activities, they are easy to propagate and wide strain selection, highly versatile and productive over other sources. Fermentation is inherently faster and more productive production compared to any other chemical process. Easy to manipulate genes. If you want more pigment production, then you can manipulate genes and you can get the high yield. Then simple and fast culturing techniques allowing continuous bioreactor operations. Structural complexity suits for industrial needs. Then bacterial pigments extracted using simple liquid-liquid extraction techniques. It minimizes the operation cost also. And cheap substrates used for bulk production. Okay, so these are the reasons why microbial pigments are nowadays they are used widely. Okay, so these are some extra examples, easy examples to remember. Okay, so if you want to uh, answer for, say, give two examples for bacteria which are known to produce pigments, then you can say either bacillus or flavobacterium. Okay, then for another question that was give examples of pigments with their properties, for even that you can use these examples. Okay, so I hope this video was useful to you all. So thank you for watching, and do like my videos, share these videos with your friends, and do tell them to subscribe, and you also subscribe to my channel and hit the bell icon so that on every niche new upload you will get the notification. Okay, thank you.